thank Jenny on the call, who is our marketing and communications manager. She'll be running tech support. I want to introduce our two speakers. So first, Dr. Chris Burns. Chris is an experienced drug discovery leader, having worked in various roles in pharma, biotech, and academia for 25 years. He finished his PhD in organic chemistry at the University of Melbourne and did his postdoc in the US at Penn State before moving to Pfizer, UK. After 5 years, he returned to Australia as a research fellow at the University of Sydney before joining biotechnology company Ambry as head of chemistry. He then moved to Melbourne-based biotech Cytopia as head of medicinal chemistry and later as research director. And over this time, he led teams in the discovery of 2 anti-cancer agents that have entered clinical trials, including Momelotinib, which was recently acquired by GSK. Chris was subsequently recruited to the Walter and Eliza Hall Institute of Medical Research in Melbourne as lab head. And he's also taken on executive and leadership roles in a number of privately held biotech companies in Melbourne, including Certa Therapeutics and MicRx. Dr. Burns is the inventor on over 30 patents and co-author on over 60 scientific publications, and is a fellow of the Royal Society of Chemistry and Royal Australian Chemical Institute. Dr. Burns is currently Chief Executive Officer and Managing Director of Australian-listed biotech Amplia Therapeutics. Professor Andrew Wilks is a serial entrepreneur with over 20 years of commercial experience, following a successful academic career at the Ludwig Institute for Cancer Research. At the Ludwig, Andrew discovered and named many novel protein kinases, including the very famous JAK family of kinases, and went on to found Cytopia in 1997. Who knows, we might see a Nobel Prize in the future for his work on the JAK kinases. Cytopia is one of Australia's earliest listed drug discovery companies, and since then he has founded or co-founded 10 companies that undertake self-funded drug discovery or provide drug discovery services to the Australian drug discovery sector. In particular, he is co-founder with Dr. Xian Bu of Synthesis Medchem, a global CRO with laboratories in China and more than 250 scientists worldwide. He also co-founded The Farm, a venture discovery company that funds and manages drug discovery collaborations generated from academia. He holds an adjunct honorary professorship in the Department of Medicine, Nursing and Health Sciences at Monash, and he was awarded the Australian Biobusiness Award in 2007 and elected to the Australian Academy of Technological Sciences and Engineering in the same year. So I'm very happy to have both of you as guests, and I'm sure the audience is as well. We have nearly 100 attendees in the audience now who are very eager to get to know you both. Before we move to your talk, I just have a couple icebreaker questions for the two of you. How did you both meet?
Well, Chris probably has a better memory of this than I do. I do count it as among the most fortuitous encounters that I've ever had in my business career to meet Chris, and he was Cher to my Sonny, although that's probably the wrong Sonny and Cher, probably the wrong metaphor. But
thank you, Andrew.
We met. You tell it.
We met through an academic acquaintance we both had. I was, as you mentioned, Dennis, working at a company in Sydney, Ambry, developing a biosensor technology for high throughput screening, and was eager to move back into medicinal chemistry and drug design, and contacted, reached out to a few people, and there'd be advice to any person on the call is always keep speaking to your networks, and it was through that network that Andrew and I met, and we basically hit it off straight away. And I'd absolutely agree. It's been a great partnership. Back, I think it was before or around the year 2000. I'd suggest, probably, Andrew.
Late 99, early 2000. The thing that most impressed me about Chris was that he's actually mentioned, I think, in the 100th best guitar bands in Australia or something. He used to be rhythm guitarist for a group.
That's so not true.
Great.
I won't go any further with that story.
Yeah, please don't.
Yeah, well, we'll have to hear some recordings in the near future. So how about both of you scientifically? What's your favorite molecule and why? Maybe Chris, you can start.
Well, obviously, other than Momelotinib, which we're speaking about today, I think probably, if I'm brutally honest, it's something really simple like salbutamol. And that's a beta agonist, for those who don't know, for rapid relief of broncho congestion in asthma. And as a asthmatic as a child, I'm old enough to remember what it was like before salbutamol was on the market. And it's a really elegant piece of medicinal chemistry, but it also was the first drug that made me realize that that's the kind of thing I'd like to do in my career: discover drugs and treat disease.
From my point of view, imatinib. I mean, this was obviously a breakthrough molecule for all people interested in kinases. And as you'll see from my talk, kinases were kind of my thing as I was an academic. And we'd started Cytopia a year or so before imatinib came along. But it broke so many sort of trends and provided so many opportunities for the sort of thing that Chris and I have done for the rest of our lives. And I've come to great good fortune to meet many of the people who did that. Jörg Zimmerman we met, and Nick Lydon, and Alex Matter, who was famously the guy at Novartis who shepherded it through, having almost been canned on a number of occasions. So imatinib would be my favorite molecule.
Yeah, great answer. Yeah, I guess probably many of us in the audience have forgotten when kinases were thought of as undruggable.
Yes,
exactly right.
Yes.
Yeah.
Yes.
Yeah. So what are you both most excited about in drug discovery today? Chris?
Look, it's hard. I think as a chemist, a small molecule drug discovery person, it's hard not to get excited about the work in proximity induced activities like PROTACs and the like. I think that's just an incredible area, and how it's just exploded. Where I think the community's really appreciated the opportunities there. And I'm really excited to see that work develop further. And my prediction would be that there's a Nobel Prize in PROTACs without a doubt. So to me, I think that's a really exciting area. And I'm really excited to see how those drugs that are currently in the clinic develop, but also thinking about new modalities of bringing biological molecules together and the opportunities that affords.
Yeah, absolutely. Andrew?
It might be controversial, but I guess AI and its involvement in. So, I mean, I have a very checkered history in computational or involvement in computation. As a biologist, I come to it from the target point of view, and it's you're either a fool or a genius with computational chemistry, and that's amplified even more with AI. I mean, it literally has a mind of its own in order to sort of to develop the molecules, and we've been dabbling in this, and occasionally we have it's from a single screen nanomolar molecules, and from other complete and utter misses and tragic disappointments. So it's starting to refine itself. It's starting to become more important. And as a biologist, and I've never actually done chemistry in any sort of tertiary level institution, I do very much value the contribution that the real med chemists make. But AI is a good tool for the likes of me. So.
Yeah, absolutely. That's not controversial at all, actually. In one of our recent webinars, we polled the audience on areas that they found most exciting, and AI was number one, and targeted protein degradation was number two. So very, very good choice. Yeah. Have either of you played around with the new AI tools like ChatGPT or OpenAI or anything like that?
Yes, afraid I have. In fact, I was vain enough to type in the words "who discovered the Jack family of kinases," and was led to 3 or 4 completely spurious leads. So it's got a long way to go. So, but that's only my vanity. So,
nice.
I'm not played with it yet, but I think it's a strange world we're entering, where universities, at least in Australia, I'm sure elsewhere in the world, are now thinking of going back to handwritten essays and the like, because they don't trust people using AI to write essays for them, which is very, very odd, bizarre.
Great. Well, yeah, last question before I turn it over to both of you. You've both had very successful careers in drug discovery. If you could each go back in time to when you started your careers and give yourselves any advice, what would it be?
Do you want to take that one first, Andrew? I can think of, while you do answering, I'll think of an answer.
So, I've had very much a two-sided career, almost an equal proportion, an academic career and a kind of applied or translational career. And I was the most snooty and sort of self-indulgent academic for the longest time, very much poo-pooed this notion of translation. All of the experiments I did were for the greater knowledge and stuff. And I regret now having had that stance. I wish I'd become more translational early on. I mean, you'll see from some of the slides, I had an interest in Drosophila. I had an interest in zebrafish. I was incredibly self-indulgent, very interesting, wonderful, fascinating, sort of enthralling career. But I arrived quite late at the idea that you could actually make a difference with a more of a translational mindset. And I kind of mildly regret that I didn't come to that epiphany somewhat earlier than I actually did.
That's a good answer. I would probably say to always seek out and work with people from different backgrounds. And I mentioned earlier on, and in the introduction, you mentioned, Dennis, I worked on a company developing biosensor technology which is definitely out of the normal training for a medicinal chemist. But I learned an awful lot in that program that I've been able to call on that knowledge in drug discovery and working obviously at the interface of chemistry and biology and working with biologists and other technologists. So I find really fascinating, and probably would have liked to have done more of that in the early phase of my career to sort of build out my knowledge. So that probably be one piece of advice I'd give myself.
Thank you both. Yeah, beautiful answers. So thank you for reflecting, and I will let, I guess, Chris take it over.
Yeah, I'll see if I can share that. So the title of the talk has changed somewhat, but hopefully it's not an issue for everybody. Andrew, I'm going to hand it over to you. And
so this is a paradigm of Chris and my relationship. Chris does all the work, and I do all of the show pony stuff. So he's going to actually do the transitions, and hopefully we're going to find our way through this presentation. Next slide, Chris.
Okay, so oh, there we are. That was supposed to be rotating. My apologies.
That's okay. Next slide. So when I'm actually from Liverpool in the UK, and arrived in Australia in 1983 to be introduced to the Don. Obviously not, fortunately not this one, but this one. This is Professor Don Metcalf, one of Australia's greatest clinical scientists, and the father of modern hematology. And literally, as I walked through the door at the Ludwig Institute when I arrived, I was confronted with this terrifying individual who was the most, actually spawned an entire generation here in Australia and across the world of people who were interested in the molecular mechanics of how all of the cells in our blood system are actually set up. And his masterpiece is on the next slide. This is a diagram that you would have seen, I would imagine, in your work many times. All of this work actually stemmed from massive contributions that Don had discovered during his career. Very nearly won the Nobel Prize, certainly won the Lasker Prize. This is the sort of choreography of the hemopoietic system. He he discovered that there were hematopoietic stem cells, cells with pluripotency that were able to actually contribute to all compartments in the hematopoietic system, that there were committed progenitor cells, cells that had somewhat limited potential, so that cells that would become lymphatic cells, cells that would become red blood cells, cells that would become only cells of the white blood cell compartment. And more than that, having demonstrated this choreography, next slide, he showed that there were in fact growth factors that coordinated this whole process, that were specific to certain cellular fates that these stem cells would actually direct themselves. And so, having shown the choreography of the cells, and having shown that these molecules worked, I kind of walked in to a situation where this was now starting to become a problem of molecular biology. And as a molecular biologist, as someone who could do all the cloning stuff, I came in and was invited to participate in what was at that time the most exciting cell biology problem that was imaginable, and that was the coordination of the hematopoietic blood system, all of which Don had coordinated. Next slide. And so my brief was to try and clone the cellular receptors for these cytokines, the cellular receptors for these molecules that coordinated the differentiation and development of committed progenitor cells through to those mature cells of the hematopoietic compartment. Of particular interest at that time was granulocytes and macrophages, and how they emerged from the transformation of these committed progenitor cells to the more mature phenotype. And the assumption was that there were cellular receptors for the cytokines that had been discovered, and that they, in fact, were part and parcel of how those cells actually achieved that differentiation process. Next slide. So at that time, and this was around 1988, the kinome was a much smaller place, that there were probably 25 or 30 protein tyrosine kinases that had been discovered, many of which were either genes that had been taken up by retroviruses, and so they were transformative in their properties. They became the transforming genes or oncogenes of these retroviruses. And then there was another set of molecules of a similar ilk that had been discovered as being cellular receptors. So cellular receptors for EGF, cellular receptors for MCSF, and so forth. And so the idea struck me, next slide, that there was a great possibility that my much sought after cellular receptors for the cytokines, so cytokine receptors, might be tyrosine kinases as well. And so I started to develop a bunch of techniques that were aimed at discovering new tyrosine kinases, with the possibility that within that pool of new tyrosine kinases, I would discover the cytokine receptors, because they were most likely to be part of that pool of new kinases. Next slide. And go back. So what I basically did was, noting that all of these kinases had within their kinase domain areas of high commonality. So you can see here this sequence VHRDL and this SDVWSF. They were in all kinases. They, in fact, when we finally figured this out, they fold up to form the catalytic site of these kinases. They were highly conserved, and so you can actually make, and PCR had literally just been invented. I'd seen the paper from Seiki in Science, and then had developed this technique within a week, that you could make degenerated oligonucleotide against these highly conserved regions, point them at each other for a PCR reaction, and that you would see in between those regions of high homology, you would see sort of diagnostic or signature regions that would tell you what the receptor was, what the kinase actually was. And so I did this. The paper was published at the bottom there in 1989. Next slide. And there it is. And this is an original gel. So doing this on cells of hematopoietic origin, I've got this fat little PCR band, exactly the right size that would be predicted from the amplification of a tyrosine kinase, exactly the distance, about 300 bases between the two oligonucleotides, and cloned them, and spent, if I remember correctly, about 2 or 3 weeks just harvesting these brand new kinases that came out from this screen. And it was an absolute torrent of new kinases. Next slide. Some of them were the sort of thing I was interested in. We now know what they are, but the VEGF receptor came out of this, TIE2, the FGF receptor, RIC, which is part of the non-canonical Wnt pathway, and then a bunch of intracellular kinases, and then 2 of them, FD17 and FD22, that were odd, different, quite bizarre in some respects. And so, next slide. Those that looked like kinases, receptor kinases, they would have the signature kinase domain, and they would have the transmembrane region. Next slide. VEGF receptor, TIE2 and stuff, they were clearly receptors. And then there were these, as I cloned out the full length sequence of these other ones, FD17 and FD22, became clear that they were not kinases. They had no transmembrane region. They were not what I was looking for. And so I called them just another kinase 1, JAK1, and just another kinase 2, JAK2. And that's where the name JAK actually came from. Now, next slide. When I published this, and I think I published the first paper in 91, I realized I couldn't use that as the acronym, as the way of generating the acronym JAK. And so I had to find another way to actually justify that acronym. And so what I did was, I've been doing a lot of reading of a chap called Arthur Kessler, and he had this notion of the Janus principle. Janus, I learned, was the Roman god of doorways and would look out and would look in. So looked in both directions, had 2 faces essentially. And metaphorically speaking, with 2 kinase domains, I kind of justified the whole acronym as the Janus kinases. They had 2 kinase domains. They look like the Roman god Janus. And in fact, this is the structure of them, and surprisingly good description of what they actually look like when they're put together like this. And so I was able to justify the acronym JAK by now using it, the word Janus kinase as the way of justifying it. And so that's where the proteins came from. That's where the name came from. And I guess the rest is history. Next slide. So immediately, having discovered these molecules, we started to try and figure out what it was they were involved in, where they became important as part of this intracellular signaling pathways. And we immediately came across a number of other people who are on a similar journey. So you'll see Jim Donnell, who've been working on the stats, George Stark and Ian Kerr, who've been creating interferon mutants. And so there was this kind of Manhattan project that kind of emerged from all of this. And we provided our full-length JAK molecules, and Jim provided his full-length STAT molecules. And these mutants, next slide, that Kerr and Stark had created, which were deficient in the ability to translate the extracellular stimulation of the receptors with interferon gamma and interferon alpha and beta, were unable to do the things that interferon normally does to cells. And they were mutant in various key elements in the pathway. And so we provided our JAK molecules, TYK2, which was discovered completely independently by Krolowski's group. He participated as well. And we started to elaborate a pathway that started with the receptor, had the JAKs associated with the receptor, phosphorylated the STAT proteins who created these dimer transcription factors that actually activated whole programs of gene activation within the cell. And so the JAK-STAT pathway was put together through the combination of these interferon signaling mutants, provision of JAK mutants and STAT mutants. Next slide. Next slide. Just work your way through this. Okay. Next slide. And then through doing, cellular biochemistry, demonstrating that, in fact, downstream of interferon alpha, interferon beta, you could demonstrate phosphorylation of JAK1, for example. So that's a hallmark of the activation of these kinases. And so you could see phosphorylation of STAT1 and STAT2 and STAT3. So the whole process was put together through this complementation approach, through the mutants that Kerr and Stark had done, and then the cellular biochemistry. And as I said in my introduction, I mean, one of the things that I would most like to have done was realize that there was a real opportunity to have an impact on humans here, because what I did was distract myself by going into model systems. But this pathway, this really elegant and very direct pathway, the JAK-STAT pathway from cellular receptor to the genome to the activation of genes, was elaborated in that process. And just interestingly, the SOX pathway, which is a reset on the JAK-STAT pathway, SOX was actually discovered in Melbourne as well by Doug Hilton and his colleagues. So this is a very much an Australian pathway. Next slide. So I want to start to make the transition to talking about momelotinib now, but really important is the contribution, I think, that some of these model systems showed the potential for. And again, as I said, I loved academia. Drosophila geneticists have all the fun. The Dorothy enhancer has tin man binding sites and drives hopscotch-induced tumor formation, I think, is a masterpiece in all article titles. And I was very jealous of how these guys were able to do this sort of stuff. Next slide. But the importance of this was that I got a very heated email from Norbert Perryman and Rich Benari in Boston, and what they discovered was the Drosophila version of JAK, and it had two different properties. It was, as a null mutant, these animals, or these flies, had a deficiency in their ability to segment, and so it's clearly an important part of the differentiated process there, these JAK, the Drosophila JAK molecule. But very importantly, next slide, it was also, I think, an example of the only example of a dominant oncogene in Drosophila, and these mutants of hypermorphic mutants of Drosophila JAK cause these melanotic tumors to form in the embryos as they developed, which is kind of intriguing. But most importantly, next slide, when they found out where these things were, and you can see T24 here, that's smack bang in the middle of the JH2 domain. And why is that interesting? Well, next slide. As we subsequently found out, and as you'll see, most of the development of momelotinib has been based on this observation, that there is a hypermorphic allele of JAK2, which drives these myeloproliferative disorders that characteristically in PRV and ET, next slide, where there are too many red cells and too many platelets formed, that this hypermorphic allele leads to ultimately this terrible disease, idiopathic myelofibrosis. And the notion was that, quite apart from the fact that these JAKs were so important to a whole bunch of processes that were potentially involved in inflammatory diseases, we had yet to figure all that out. There was a single gene mutation that caused a disease that was well worth the development of JAK2 specific inhibitors. And so that's kind of what drove our thinking there. Next slide. So this is that disease, about 80,000 people. It will become very important as Chris talks in a moment, but it was already foreshadowed in the mid-90s that this would happen through the demonstration that there was a hypermorphic allele of JAK in Drosophila that caused cancer in that organism. Next slide. And this idiopathic myelofibrosis will again be very much the justification of the development of momelotinib, but it's the end stage of that disease where you get this extramedullary hemopoiesis and the splenomegaly and so forth. Next slide. So I did have that epiphany in the mid-90s. I did decide that it was of great interest to do the academic thing, but a real impact could be made if you could apply the understanding, the learnings. In fact, I had patents on the JAK kinases at that time, or the actual existence of the proteins, that you could screen them and so forth. And it was either a midlife crisis or some other decision I can't quite figure out, but I decided I was going to leave academia and pursue the translation of all the information that we'd gathered on these JAK-STAT pathway and the patents on the JAK kinases, and start a company. And the company was called Cytopia. And as I say, I left the Ludwig Institute in '97 to start the company. And next slide, immediately fell on hard times, which you'll see next slide. That symbol is that's what my wife calls the baked bean years, because we went through quite a traumatic time trying to set up Cytopia. I had some seed money, and we were using that seed money to go out and find investors. Unfortunately, having left the Ludwig, they also pulled the seed money, and so I had no visible form of income for quite a considerable period of time. In fact, I would claim that at that time I was a chess professional for a period, in as much as the only income I had was I came 6th in the Australian Lightning Chess Championship and won $123. And so it was really quite tough times as we tried to start this company. But then, next slide, Medica Holdings came along and they gave us some cash. And as I said, to my great good fortune, I came across the good Dr. Burns here, who very much took the lead from the original screens that we'd done. And it's a great tribute to his contribution that these molecules, lexibulin and CYT387 or Momelotinib, were discovered. And I'm going to give it to Chris now, so he can carry on telling the whole story of what happened from there.
Thanks, Andrew. Yeah, so as we mentioned, Andrew and I connected in around the year 2000 after Andrew had established Cytopia and really built up, I think, and it's the strength, I think, of the company at the time was that we had really deep knowledge of the way the JAKs worked. We had good screening systems, and so we were able to get into the chemistry very quickly. Didn't mean that we didn't go down a couple of blind alleys, but nonetheless, the outcome of all that work was the discovery of this compound CYT387, now called Momelotinib, which the structure is shown here. So very much that kind of first generation kinase inhibitor based on a pyrimidine scaffold. Just to remind everybody, again, this was really driven by this identification in 2005 that there was this activated mutation in JAK2. Prior to that, we were still interested in JAK inhibitors because of their involvement in a range of cytokine signaling. But it was this identification that really got us excited, and indeed, I think a lot of other companies. And we've already spoken about the incidence of that mutation in various diseases. We'd been working in compounds of this type for a little while, and we had a number of different libraries. We were working with a talented group of modelers, and basically, we identified quite early on that compounds of this type were preferential JAK2 inhibitors. And what I'm showing here is a collection of data. Each dot is the data for a compound. And what this assay is a cell proliferation assay where you're comparing an isogenic cell line, one driven by JAK2 and one driven by JAK3. So everything else is equal. And what you can see is we've got most of these compounds sit on this side of the red line. So it's sitting up here, indicating that they're more potent on JAK2 than JAK3. And that was really important for us because our main concern in targeting JAK2 was to get selectivity over JAK3, because JAK3 is downstream of IL-2 and IL-4. There's clear genetic evidence that inhibiting JAK3 or knocking out JAK3 leads to immune suppression. And we wanted to make sure that our compounds weren't doing that. And indeed, that was really pleasing, but somewhat surprising, given the fact that there's such close homology, particularly in the ATP binding site between JAK2, which is at the top here, and JAK3, which is down here. And I've just circled the 2 amino acids that are different in that ATP binding site. So a serine to a cysteine and a glycine to an alanine. You know, seeking selectivity for JAK2, therefore, you know, was very pleasing. As an aside, I will say that we took some of our weaker JAK2 inhibitors and actually modified them to target the cysteine of JAK3. So added a warhead like an acrylamide onto those compounds to get selective JAK3 inhibitors. And this was done in the early 2000s, a long time before ibrutinib and a long time before companies like Principia were doing this. In fact, the only people who were thinking about irreversible kinase inhibitors at that time was the group at Park Davis, and they were working closely with Bill Denny and his colleagues at the University of Auckland. So this is work we were doing on targeting JAK3, totally separate to this talk. And that led to a long collaboration with Novartis, ended at the time of the global financial crisis. Unfortunately, it's a bit of a crunch, obviously in collaborative opportunities. But that work lives on in a whole range of now irreversible inhibitors, and particularly I acknowledge the work that Pfizer have done recently with ritlecitinib, which is now being developed in alopecia and some other immune diseases. But that's really a bit of an aside. I wanted to put in a bit of chemistry, always taught to do that at Pfizer. When we're talking medchem, it is chemistry, of course. Pretty straightforward chemistry here, Suzuki or cross-coupling type chemistry to put on the aryl group, and then nucleophilic displacement, either with palladium catalysis or just classical displacement reactions. And so what we did, we knew that the aniline portion, based on our modeling, was pointing out into solvent, and so this gave us an opportunity to improve and modulate solubility, as well as trying to pick up some binding interactions. This right-hand side from early work seemed to be reasonably potent in the JAKs. So we made a series of anilines. I'm just showing a couple of examples here. So we have biochemical potency on JAK2 and on JAK3. So we're seeing really good selectivity in the biochemical assay, and also that translated generally into potency and selectivity in a cellular readout. The morpholino aniline portion here down the bottom was the best of the bunch, and so we kept that constant, and then investigated the aryl group on the right hand side. Again, we made a lot of compounds here, but of the ones we were making at that time, this phenol actually came out as being really interesting, and a lot of cellular assays. You know, this compound was really very exciting. We got a crystal structure of it. It bound exactly as we thought. So we had this hydrogen bonding dyad here to the hinge region of the JAKs. The problem with this compound is probably everyone in the audience has noted, we've got a phenol here, which was of course rapidly glucuronidated, leading to a very short half-life in vivo. Spend a lot of time trying to modulate this basicity or acidity of this compound, I should say, or modulate its glucuronidation, which we could never achieve actually. Steric encumbrance, twisting the molecule, putting it out of plane, which was something that had been done in other programs. And so we came up against a bit of a brick wall. This was an exciting compound in a number of our assays, but obviously could be used in vivo. At that point, we were working with a very talented group of people at Monash University, which is in Melbourne. Isabelle Lucette, who was the first person to crystallize JAK2 and get a structure of JAK2. And she crystallized this compound, which is now known as tofacitinib, CP690550 at the time, a so-called JAK1, JAK3 inhibitor, but it in fact is reasonably potent on JAK2. And what was really interesting to us is here's the hinge region. So you had this hinge binding head group, but we saw that the back end of the molecule, if you like, and particularly this nitrile, was pointing up into the what's called the glycine-rich loop, undertaking a hydrogen bond with the backbone there. And we looked at that and said, geez, wouldn't it be nice if we had one of these in our molecule? So pretty much went away straight away once we had that crystal structure and made a whole series of nitrile substituted right-hand sides of the molecule as they're drawn here. And again, I've just got 4 examples. And what you can see at the meta position, it didn't do much. The para position, we saw much better activity, and indeed this compound at the bottom here is CYT387, so 10-fold selectivity in the biochemical assay and about 5-fold in the cellular assay that we were working with at the time. Hasten to add, we made many more compounds than what I'm showing you here, and CYT387 was always used as our standard, our benchmark, and we never improved upon that molecule across all the different assays and tests we put it through. CYT387 had a unique balance of activities. I've got some of the collected data here. So selectivity was equipotent on JAK1 in biochemical assays. Looks equipotent on TYK2, but when you actually run a KD, it's actually a lot weaker on TYK2. And JAK3 I mentioned before, also potent when you have the V617F mutation as well. And then in cell lines that are driven by JAK2, we see good potency, sometimes in the low 100 nanomolars, a lot weaker on cells driven by JAK3, and totally inactive on cells like K562 that obviously are non-JAK dependent. And looking at downstream effectors, Andrew mentioned the phosphorylation of the stats. We can see that this molecule dose dependently inhibits stat phosphorylation in a number of different cell types. Probably the really critical aspect of CYT387, other than the activity I've just mentioned, was it really does have exceptional ADME properties. In the rat, 100% bioavailability. In fact, in some of the studies, it was greater than 100, which was amusing. Good half-life. In the dog, the bioavailability was 50%, and I think that's pretty similar to what's seen in humans. No problems with permeability, CYP inhibition, hERG and the like. And so this molecule was chosen to move forward into a number of models. We worked with a group at Oregon Health Sciences University, Mike Denninger. Mike moved on from there now, but Mike had a model of myeloproliferative diseases. So lethally irradiated mice were basically transfected with a V617F mutant JAK2 and developed essentially a myeloproliferative disease where you had increased red and white blood cells and enlarged spleen. And what we showed in this model was that we saw for animals dosed with CYT387 a dose-dependent reduction in spleen size. So this is spleen size at the vehicle-treated animals. This is 25, 50 mg per kg a day of drug. That's what a normal mouse's spleen looks like in the yellow. So we see a really appropriate reduction on spleen size. We also saw that in blood cell counts. I've got the white blood cell count here, but as soon as we started therapy, when that blood cell count was quite high, we brought it back into a normal range, which is shown here in gray, and again in a dose-dependent manner. Vehicle-treated mice have this much larger numbers of white blood cells. And similarly, you see an anti-inflammatory effect as well. Not so much dose dependent here, but we see a significant reduction of a range of inflammatory cytokines compared to vehicle-treated animals, shown down here on the bottom right. So all of that gave us confidence to move this forward into the clinic. There's a study that was initiated in 2009 that we began in at Cytopia, working with a team at the Mayo Clinic in the US. And I think this was really one of those few times in my career where you get data that is profoundly I guess moving and really makes you think that you're onto something. First of all, the drug was well tolerated. There were no significant toxicities. But this was given to elderly patients who had myelofibrosis. With myelofibrosis, you get this enlarged spleen. Andrew showed the size before, the spleen size, how that leads to a lot of discomfort. But you also have a range of constitutional symptoms: night sweats, bone pain, chronic itchiness, fever. Really, prior to the JAK inhibitors coming online, this was really a terrible disease. Average lifespan was about 5 years. And we got this data pretty quickly after patients were starting to take the drug, where this is percentage improvement, where this is patient-reported improvement, where they see significant improvement at both doses in all these constitutional symptoms. And we were getting stories of people that had been bedridden, were back out, living their life, enjoying retirement or getting back to work. And so that was really wonderful to hear that news. And what was surprising and exciting too at the same time is we also saw that a lot of these patients who had been anemic were no longer showing anemia, or at least a significant reduction in their anemia. And indeed, transfusion dependent patients were becoming transfusion independent. And that obviously, again, led to an improvement in quality of life. And as the studies went on, and this is data that came out of subsequent clinical studies, blue here is Momelotinib, and you can see that patients' hemoglobin always stays above this kind of threshold of where people need transfusion. Other JAK inhibitors like ruxolitinib led to people becoming more anemic and requiring transfusions as soon as they switched to Momelotinib. Of course, that anemia effect of Momelotinib was seen, and the anemia went away. That's not only important in terms of quality of life, but there's a lot of evidence showing that patients who are anemic actually with myelofibrosis have a worse outcome, lower survival. And so addressing anemia on top of these constitutional symptoms, the splenomegaly, is a really profound benefit of Momelotinib, and something that was seen as very, very exciting. So the current status, I'm conscious of the time, so I'll wind it up in a moment, Dennis, is that the drug, an NDA has been filed, the PDUFA date's been set for 2023. It'll be initially a second-line treatment for a relapsed or refractory JAK inhibitor patient. And this is an image that came from Sierra Oncology, who are developing the drug up until recently. It inhibits JAK1 and JAK2, which addresses the hyperactive JAK signaling. But in subsequent work that I initiated, but it was really driven by Igor Thiel at Innsbruck and Andy Whitney at Gilead at the time, was to identify that Momelotinib, despite being a very clean kinase inhibitor, actually inhibited one other kinase, ACVR, and it was through inhibition of that affected iron metabolism and homeostasis, leading to this beneficial effect in anemia. So it's one thing I always say, it's You need a little bit of luck in drug discovery, and this was definitely a bit of luck that we had with the development of this agent. So the team that we work with, or this discovery team was all based in Melbourne, though we worked with a lot of labs around the world doing the translational arm of this, and including the clinical study, the phase 1 studies at the Mayo Clinic. So obviously want to thank all those people. Before we finish, I wanted to just take you through a really interesting aspect of this story, that Cytopia got to 2009. We had 2 compounds in the clinic, one of those being Momelotinib, and couldn't raise funds. It was the height of the global financial crisis, and the company was ultimately acquired for 10 million, this is all US dollars, by YM Biosciences, a Canadian company. They continued the Momelotinib trial for 2 years, kept on putting money into the trial, but nothing else had changed really. They were acquired 2 years later for $510 million, so that's quite a nice uptick, by Gilead. And Gilead did some really nice work on drug product optimization and CMC optimization that sort of refreshed the IP. Had a bit of a phase 3, well, they ran the phase 3 trial, but it was mixed success, and ultimately sold the IP plus some royalties to a group of scientists at Sierra Oncology for around $3 million. Sierra then completed or reran the phase 3 trial with close engagement with the FDA, and it was that work that led ultimately to the acquisition by GSK for $1.9 billion. So that's quite interesting tale of the value of this asset over time. And GSK are now in the process of working this up to release, hopefully later this year. So I'll stop on that point. Though I don't know, Andrew, if you wanted to make any further comments, but happy to leave it there.
I was having a lot of fun until you came up with that slide, Chris. I mean, that's quite harrowing how the transition for 10 mil and its subsequent acquisition at 1.9 billion, the multiples there are quite substantial. Neither you nor I suspect, got our fair dues out of that process, but hopefully we get the glory, which is. Someone
asked me what kind of car I drive, and I said I still drive a Toyota Yaris. So nothing. Nothing has led to me buying the Ferrari, unfortunately. So we'll leave it there, Dennis.
All right. Well, thank you both so much for sharing your story. It's absolutely fascinating. For some context for the audience, the way we got in touch with Andrew was we had put out an article on the GSK acquisition of Sierra Oncology. And when we were digging through the history of the molecule, we discovered, wow, there was this Australian biotech called Cytopia that was involved, and it traced all the way back to the discoverer of the JAK enzymes, which was truly fascinating. So thank you. I see there's already a comment in the audience saying, wonderful talk, wow. And we have a couple questions for you. Okay, so first question from the audience is, can you comment on the preclinical safety studies? What were the expected TIs, and was there a target organ identified?
Yeah, I probably don't want to go into too much detail because I'm not too sure what is ultimately in the public domain. In terms of preclinical modeling, the drug was ostensibly seen as safe. We hadn't observed any of the kind of toxicities that have been dogging JAK inhibitors more recently. So we were confident that going into this patient population that the drug would be safe and well tolerated, and the data actually supported that. Andrew, I don't know if you want to make a comment on that too.
It does raise a very important point, because it's not just the V617F generated diseases where these molecules are useful. I mean, in a whole bunch of chronic inflammatory disorders, so rheumatoid arthritis and IBD and stuff, and famously, there was the Gilead/Galapagos molecule, that it was approved in Europe but not in the US, and so there's a few black box warnings out there, and the biology is so complex, and the blend of JAK1 versus JAK2 versus JAK3 is apparently quite an important distinction between a lot of these molecules. And you add onto that our observation, or Chris's observation in particular, that there were other collateral activities that actually mitigated some of the potential issues that these molecules can cause. We still genuinely don't have a good handle on where that sweet spot of efficacy and off target or on target toxicities might lie. I think with this molecule have fallen into a quite an interesting sort of overlap of that Venn diagram, where efficacy and safety seem to be sitting elegantly side by side, and that's both a tribute to Chris and his team and what they've generated, but also that serendipitous observation. But we still, I don't think, have a full handle on that where the perfect molecules might sit that would not give rise to some of these potential off-target activities. I think we're very fortunate to have gotten where we were.
Yeah, thank you for that. Yeah, it's a very impressive drug profile that is very unexpected, especially the anemia properties you've mentioned, but hopefully will be very valuable for patients. Dennis Koester also has a follow-up question to that. Did you, at the time in preclinical studies, did you look at spleen exposure in particular, or blood to plasma ratios of the drug?
Not spleen exposure, at least as far as I can remember. Blood to plasma was as predicted. There was no particular compartmentalization of the molecule. So, but we didn't look at that. May have been done subsequently of exposure in the spleen.
All right. Zhonghua Pei in the audience has another set of questions. He asks, for the inhibition of off-target ACBR1, it was serendipitous, but do you more fully understand the activity of the drug on that target now?
It was absolutely serendipitous. As I said, a lot of this work was driven when the drug was with Gilead. It is still very much an ATP binding site inhibitor in that particular kinase, other than that, I don't think I can say much more. Really, it is just an off-target activity that just seems to work. So yeah, probably leave it at that.
I'm not sure if you can comment on this, but at the time that at the time that Gilead had acquired it, did they know about that activity, or was this discovered after they had bought the molecule?
No, we knew it had the anemia effect, and in fact, the history of that discovery was, we'd been thinking about opportunities outside of myeloproliferative diseases for JAK2 inhibitors, and Igor Fuel emailed me out of the blue and said, I have a model of chronic anemia of disease. We think that might be IL-2 driven, or IL-3 driven and IL-6 driven, so JAK2. Would you be willing to work with us? And it sounded like a really interesting opportunity, and we started working with Igor and got some really exciting data at about the time we were in advanced discussions with Gilead. So they had seen some of the promising data from the clinic. We were getting this additional data from preclinical models, well-defined preclinical models, and that obviously was then really picked up by them and chased down to this particular activity.
Gotcha. Okay, so we have one last question for time. Zhonghua also asks, are you at liberty to say why Gilead ultimately sold the molecule or gave up on it? That $3 million sale price is pretty shocking in hindsight.
Yeah. Look, I'll give you my perspective. It's totally my perspective. But it's an interesting sort of insight, I think, into the way large biotech works with small biotech and everyone will say this when you're doing business development, a lot of it is about personal relationships and internal champions. We had a really good relationship with a number of the scientists and key people at Gilead. And just by the normal churn of the industry, those people left and moved on to other roles. And I think we lost a few internal champions, unfortunately. And Gilead were obviously doing a lot of exciting things in other areas. Momelotinib didn't read out as well as they would have liked in the phase 3 trial. And given the way they structured the phase 3 trial, the anemia benefit wasn't able to be factored into a registration. There's probably a couple of things there.
Gotcha. Well,
I think Chris is spot on there, and they blundered a little bit in the way they constructed their scoring function for the outcome of the clinical trial, and so it didn't quite reach significance. And maybe it was embarrassment, or maybe it was because they'd lost the people who were driving it. They offloaded it, and it's a tribute to the Sierra team, and that included Chris. I know for quite some time that they kept running with that ball, and I'm very grateful that they did, because this is the best molecule in that setting, and so we're kind of excited about that. I don't know. There's a couple more questions, Dennis, but
yes.
I know we're running out of time. Can I just say two things? Firstly, Amanda, I have worked on kinases in malaria. If you can get my email address to Amanda, I'm more than happy to talk about that. And Patrick, I know I owe you an email, and I can see you asking me a question that I will respond in the near future.
Fantastic. Well, thank you both so much. This is a tremendously insightful and interesting talk. I see comments from the audience reflecting that as well. Dennis Gosser says, thank you for delivering such an impactful story with great humor. I know one of the hardest things about giving a webinar is not being able to see the audience's responses, but I can assure you we were all following along with great interest. So thank you very much. So there were 120 participants at the peak.